So I think we can start. So it's really our great pleasure to welcome all of you to this new series, new season of the European Webinar Microglia Series. You might know this was the series of seminars launched by Guy Brown, that is here in the audience. Guy Brown, University of Cambridge, together with Tim Burkle and Claire Butler. I think that the very first talk was in November 2020. And it was such a great initiative that in fact was having a great success. And now it's our pleasure to take over the coordination. And when I say our pleasure, I refer to two teams. One is in Spain, Amanda Sierra, Achucarro Basque Center for Neuroscience, together with Federico Soria, she's here in the audience, and Marta Pereira. And the other part of the team is at UNIL, University of Lausanne, and it's me, Rosa Paulicelli, together with Anne-Claire Compagnon, Fanny Martineau, and Giorgio Corsi. So as you know, this is a series of microglia focus meeting, will be a monthly, the month appointment, and we try to keep the tradition, like Guy launched, having once a month, whenever possible, the first Monday of the month at 5 p.m. Central European Time. So we have already a program, and I will leave actually now the word to Anne-Claire, that will tell you a bit more about it.
Yes, so hello everybody. So speaking now for the organizer committee of the webinar. So first to say thank you to be here and supporting this initiative. So also a few months ago, we sent you a poll with questions and open suggestions about the webinar format, and also to put potential name for new invited speakers for the year to come. So also here, you were really many people to reply. So thank you again for this also. So we will take all your suggestions into account, into consideration, and we already selected some speakers you suggested from January on. Also, we would like to mention that we have a Twitter account now, so you can also follow all the updates on the Twitter. So we will put the link of the Twitter maybe in the comment section. So we will also put on Twitter and pin it on Twitter the email address of the webinar series. So for all the people who wants to register to the webinar series and the newsletter, so that you can receive the invitation also to follow the upcoming seminars. And also, finally, we want to put also on Twitter, and you will also receive on the next email the link of the recorded talks. So for sure, of the speaker who agree that to be recorded. And this will be placed on the Achucarro website. So in case you could not attend it, you could still see the webinar online. So I think that's it for all the updates. And I will let Fanny now to introduce you the speaker of today.
Thank you. So it's my great pleasure today to introduce the first speaker of the year, which is Professor Sandra Siegert. After her undergrad studies, Professor Siegert moved from Germany to Switzerland, where she started her PhD journey at FMI in Basel under the supervision of Professor Boton-Rosca. There, she focused on molecular barcoding of retinal cell types and developed a comprehensive atlas of retinal cell types, which actually made it to the cover of Nature Neuroscience. After her graduation in 2011, she started her postdoc research in Lee Whiteside's lab at MIT, where she showed that the epigenetic regulator microRNA 137 is involved in learning and memory deficits that resemble the onset of schizophrenia. In 2015, she moved back to Europe and she started her own lab at ISTA, the Institute of Science and Technology Austria, just outside of Vienna. And this is where she moved her studies to microglia biology. Professor Siegert received a number of prestigious distinctions, amongst which an EMBO fellowship and an ERC starting grant. And very recently, in 2023, she was actually promoted to tenured professor. So today she will tell us about microglia morphology and function, and we'll probably sprinkle a bit of ketamine on top of that.
So thank you very much, Fanny, for the introduction. And yeah, so let's immediately start with the talk about from microglia morphology to function, and short disclosure. So just the disclaimer. So I'm also a co-founder of Syntropic Medical, but it should not matter so much for this talk. So let's start, Emil, with the beauty of this meeting is, and thank you again for this invitation, is that I don't have to talk so much about microglia and introducing them, how beautiful they are and how they are nicely spreading across the brain, and that they have a wonderful morphology distributing throughout the entire brain. Okay. We know that microglia are constantly scanning their environment, and here, for example, one of the in vivo recordings we have been performing, and we also know that they are sensing their local environment. So microglia are like around the neurons, and they're touching and interacting with the nervous system extensively. We know that they express receptors of all kinds, from adenosine to purinergic receptors. And we also know that they are extensively interacting with the different synaptic sites. Like here, for example, we are showing here the different postsynaptic sites, and you see how microglia are interacting at the different positions here. So overall, we are mostly looking always on this beautiful side of where our immunostaining is showing us the fluorescence intensity. However, we never went in and dive into the deepness of the darkness, which we could call the dark matter, or where we say this is the extracellular matrix space. And this is actually a shame, because we have a lot of insights that we can gain in looking into this kind of space. So the roadmap of my talk is to first give a short introduction about the extracellular matrix, which I assume will be rather also a new topic for most of you, as it was for me like a couple of years ago. Then I will introduce ketamine anesthesia, microglia and plasticity, and what we have learned so far in my group about these aspects. I will give a short intermezzo about microglia morphology, open ketamine exposure, and then finally we'll introduce some of our unpublished data about how does sex matter in this context. Let's go now first starting into the introduction of the extracellular space and the extracellular matrix. Extracellular space, it's actually not empty at all, as we would assume. So here is like a picture from our collaborator in group in Johann Danzel, who does like the like a staining in which she stains everything which is in the extracellular space. So he makes like a contrast image. And what is really beautiful here to see, so we have here, he had the synapses stained, and here you can see in the circle around how microglia is going through this extracellular space. That means the microglia, we know they are very active with their processes. They also expresses a lot of molecules related to extracellular modulation, MMPs, like matrix mid-pyeloproteinases or ADAMs. So all factors that allows them to navigate through the extracellular space. And how does the extracellular space look is actually representing? We have all neurons, so you have to think about that all neurons are surrounded by a glycocalyx. It's a kind of a coat of carbohydrates that is composition of glycoproteins, glycolipids, and proteoglycans. And they're forming a net structure and keeping the neurons and astrocytes or similar cells in shape. And we often underestimate the importance of the extracellular matrix, even so it has been shown to have very relevant functions. So one are cell-related, for example, like organizational structures. They can protect for certain environments. They can control navigation of receptors in space, but also they can store certain factors within these compartments, and in this way prevent, for example, the diffusion of certain molecules from one cell to the other. So overall, the ECM has an important function, and if we specifically look in the central nervous system, we can find like 3 compartments of ECMs that are commonly described. So for example, here you can have the blood vessel with the basal membrane and the endothelial cells, where you find like collagen, CSPG, and fibronectins forming a mesh around it. Or between neurons, you find like a combination with tenascin, hyaluron, collagen, and CSPG. Or a very specific and defined matrix, which is called the perineural net, which is around a certain population of cells. And this perineural net is a rigid, unique, lattice-like structure that is mostly around the cell body, but can also then spread out to the different axonal and neuronal processes. Here's, for example, one of the stainings we have been performing. So you can use this antibody, WFA, Wisteria fluoribundine, that labels this perineural net. And here we used parvalbumin, and you can very nicely observe how the WFA is enshielding this parvalbumin positive neurons in the cortex. Now, you can also see how the perineural net is distributed within the mouse. So you can see it is indeed expressed in several subpopulations within the cortical areas, and also specifically highly expressed, for example, in the CA2 region of the hippocampus. And as mentioned already, this perineural net is a very compact structure and has different features. So one is like it has been clearly shown to have a physical barrier between neurons, and in this way preventing that the neurons can form new contacts. It acts as a scaffold for inhibitors, so like preventing that the receptors can trafficking along the membrane. As well, it can store inhibitory factors in this space. And this is what happens relatively at the beginning of our lab. So we were like looking at the perineural net. This is in a control condition, and then we treated our mice, and suddenly we saw the PNN gone. And the treatment we have been doing was just a repeated exposure of ketamine anesthesia, which was like a very surprising result for us. And it was so crystal clear and black and white. We said, this is something very spectacular and we should truly look into, because that is some important modulator of neuronal activity. And so this brings me to the first second part of my talk, for looking at the ketamine anesthesia, microglia, and plasticity. And that was like when we started my lab initially, we were wondering together with Alessandro which anesthesia we should use to treat our animals and to perform our surgeries. And we had to write the animal protocol, so we had to think about this approach. And typically, when you think about general anesthesia, the main features is it's drug inducible, it's reversible, completely reversible, and it composes of a behavior state of unconsciousness, amnesia, analgesia, and it also really requires immobility along with physiological stability. And especially the reversible is a critical feature to be truly justified to be a general anesthesia. And we started now planning an experiment in which we gave repeated exposure of ketamine and investigated what could be the consequences of this. So we used Normaplex 6 adult mice. We gave ketamine and analyzed 4 hours after the treatment, the perineural net after 1, 2, 3, and 6 times exposure, with 3 days time in between. And what we found initially when we look at the somatosensory cortex in the control condition or after one-time ketamine, there was not so much difference in the number of PNN-coded cells. However, after 2 times or 3 times or even then 6 times, we saw a complete loss of the perineural nets. And we quantified it. We quantified the density of the number of PNN-coded cells, and it dropped dramatically from by 80% in both sexes, in male and in female. And at this point, we were like surprised. We also looked in other brain regions like the visual cortex. And indeed, there we also found after 3-time treatment a complete loss. And interestingly, when we looked at the recovery after 3 days, 7 days, and 14 days, we saw that the PNN comes back, which is in contrast to other classical enzymatic strategies where when you once apply, the perineural net is gone for several months. At this point, we got very curious about this effect of like depletion and reopening, because it gives us an opportunity to think about how we could modulate brain network activity. So I put here now an example of visual perception, and it's a very classical example. You have 2 eyes, and if you have a cup in front of you and you will close one eye, you will see, and then switch with the other eye back and forth, you will see that your finger or your cup or whatever you have in front of you will shift left and right. And that reflects that the brain receives 2 visual inputs from the left and right, which are slightly shifted. And then in the visual cortex, this information has to be merged together to one image with the perspective of a 3D projection. And this approach of combining those images is performed, is learned by the brain in a phase which is the critical period phase and in the visual cortex, where the brain region is very sensitive for the input for both eyes. And in this time, it learns to distinguish the input comes from the left and the right eye. And it has been shown that with maturation of this perineural net, this window closes, and then the brain is not anymore able to learn like differences in the shift. And it has been shown also when you, for example, have misinformation in this time period, for example, one eye is closed at that time, you can develop a disease called amblyopia, which is pretty common in child's, and you may have seen that some of the kids are wearing this kind of eye patches. So the idea is to try to force the brain to learn that it combines these images together. But this process is very long and tedious. Now, does ketamine reopen the plasticity? Seems to offer a possibility to reopen the plasticity window. And to address this question, we said we collaborated with Mark Baer from the Picaro Institute, in which we were looking at the visual cortex, at the binocular region and contra and ipsilateral eye. We implanted electrodes, and then 2 weeks later, we gave ketamine for 3 times. Then we gave monocular deprivation for like 3 days. And then we analyzed the visual evoked potentials immediately after monocular deprivation, 1 day or 7 days. And normally this time period would be not sufficient of 3 days to allow a shift in the ocular dominance plasticity. And this is exactly what we are seeing with the saline treated condition. There is no differences in the contra and ipsilateral eye. So there is no possibility that it's normalizing and the ratio is becoming equalized. In contrast, when we look at ketamine, initially, after the 3 times treatment, we also don't see a shift occurring after the contra and ipsilateral eye. However, after the monocular deprivation, so this 3-day interference, we see a drop in the contralateral eye. And also we've seen a significant difference in the contra-epsilon ratio, and that maintains until 7 days. So suggesting to us that we have the possibility to readjust and reopen a juvenile-like plasticity window. And so at this point, we were very excited, and we're now wondering who enables this PNN loss at this point. And I don't have to tell you, it shouldn't be a surprise for you that it is the microglia, which we found to be critical involved in this process. And what we found was that, first of all, microglia as a first indicator was that we saw an upregulation of this reactivity marker, CD68, an endosomal lysosomal marker, typical upregulated in reactive microglia and not in the saline conditions. And when we then further looked into the PNN structure, we found PNN fragments, so this WFA stained PNN fragments within the CD68 within the microglia, suggesting to us that indeed that the microglia remove this PNN within PNN over time. And here's an example where we have performed ex vivo cortical slice recordings in the somatosensory cortex after 2 second exposure. Whereas in the saline condition, you see that the microglia are just like reaching out and touching around the area on screening. They, in this reactive, after 2 times ketamine, you see a massive accumulation of the perineuronal net within the microglia, and you see that the microglia are closer with the cell body to the PNN-coded cells and start to put their fragmented inside the PNN mesh and in processes and going through this tissue. And this we also quantified, so we saw an overall increase in the number of PNN volume within WFA volume within the microglia, only within 20 minutes recordings on this. So indicating indeed that the microglia have an important role in removing this. To further substantiate this, we depleted the microglia. So here's we're using the PLX5622 in a chow food, in the chow of the feeding the animals for like 1.5 weeks, and we see a decrease in the number of microglia. So we have an 80% reduction of the microglia. And when we then look at itself at the PNN coding, we see that with the ketamine anesthesia, we don't see any changes in the number of the PNN coded, which we also quantified. So when there are no microglia or significantly reduced microglia, we don't see anymore this massive PNN removal. And we also looked at other phenotypes, and we found also that a critical feature of the PNN formation is that the parvalbumin neurons are usually enshielded by the PNN. And we can, as I've shown you before with the parvalbumin neurons, we can very nicely label them with this parvalbumin staining in the WFA. And when we then looked how the distance of the microglia are to these parvalbumin neurons, you can see this is in the saline condition. We have here the parvalbumin neurons and here the microglia. They are just touching, similar as we have seen in the live video. And in a ketamine condition, you see how the cell bodies of the microglia move towards the parvalbumin neurons and kind of almost like hugging the parvalbumin neurons. And we also quantified that there is a significant reduction in the distance of the parvalbumin and microglia with the ketamine exposure. So giving us another indication that indeed the microglia are involved. Next, we were like, curious about how this process is going. We know that there is that microglia are sensing neuronal activity, and there has been studies indicating that ketamine anesthesia leads to a massive increase in like brain activity initially, and then in the recovery. So what we did is we implanted electrodes into the visual cortex, and then recorded for 30 minutes baseline recordings, and then gave deep anesthesia, ketamine, and then recorded for another 5.5 hours. And what is very nicely here to see, when we do look at the local field oscillatory frequency, we see a massive increase here. Initially, when we give ketamine, it's like an oversaturation from the 50 to the 90, and so and a high gamma activity. And that inspired us to looking back on a paper that had been recently published by my postdoc supervisor, who reported a microglia response to 40 Hertz brain oscillation, and indicated that in this frequency, using light entrainment, the microglia are taking up amyloid plaque. And we were like, this is an interesting aspect, because we look at 60 Hertz, which is another frequency, but also have a reaction on another extracellular matrix structure like the perineural net. So we were wondering whether we can recapitulate this kind of phenotype. And we built these chambers here, where we put the mouse in here in the home cage. And we gave them different light intensities. We gave them 8 Hertz, 40 Hertz, or 60 Hertz for 2 hours for 5 days, and then as analyzed the number of PNN after the last stimulation. And to our surprise, I have to say, we found a reduction in the number of PNN by almost like 40%. And we even further looked into and very selectively to 60 Hertz, and not to 40 or 8 Hertz, or to control constant light stimulation. And also what we found is like that microglia became, similarly as we have seen with the ketamine anesthesia, closer to the parvalbumin neurons again. And we found that the microglia is, as you see here, sitting on the structure and starting to disassemble the PNN, similar as we have seen with ketamine. And as we quantified with the PNN within microglia CD68. Indeed, and again, it was also truly surprising that this is only happening with the 60 Hertz and not with the 48 Hertz or with control light. So at this point we can say that repeated anesthetic ketamine seems to make our brain more plastic, because it allows us to reopen an actually closed window and allow to readjust the brain activity. And so we were very excited about this data. But we also at this point were wondering, because we had initially only looked at CD68 as a marker, because we also looked at microglia morphology at that time, because we were like thinking there must be like morphology as an important indicator. There should be something in the morphology. However, we could never see the strong differences there, and there was a lot of variability in our data set. So that's why I give you a short intermezzo here, how we solved this microglia morphology aspect, which we initially didn't see. But we thought it must exist. So this is like just an example again of the microglia morphologies we saw at different conditions. So saline and 1-time ketamine, 2 times, 3-time ketamine. So yeah, you can see with 2 and 3 times they're probably more reactive, but then to distinguish between 1-time and saline has been very challenging at this point. So we started to think about this question and how we can create a new strategy to resolve the microglia morphology, and in a way to reduce the 3D tree complexity and maintain as many information of the 3D skeleton that we have reconstructed as possible. And the strategy we came up together with our collaborator at EPFL and KTH, and it's then called Morphomics, which consists of 3 different parts, which I just will quickly go over in a nutshell. Where we have been seeing, I think the most critical part here is like we have the rooted microglia 3, and what we are doing is like simplifying this tree into a persistent barcode. And that is done in a way in which we take a function which distance from the soma and gradually going through these processes. So when the filter reaches the first terminal endpoint, we say a process is born. Then it reaches the next endpoint, it will say an orange process is born. And then we can get to a bifurcation point, and the process which was born first continues, whereas the one which born then the recent one dies. In this case, the orange one would die and would stop here as a bar, whereas the blue would continue. And in this way, we can generate an entire map of these barcodes, which we can then simplify by collapsing them as dots, individual dots together, and then can represent them into a persistence image. And those strategies allow us then to introduce statistical approaches to deal with the microglia variability of the morphology, and to use data visualization strategies like UMAP to represent our data. And to make this, we have generated an entire map. So if you would like to look into this a little bit more, you can read the paper from Gloria and Colombo and Ryan Cubero, which we have published last year. Just to give you the beauty to represent how what we can do with it now, we can generate now these maps of, for example, like the somatosensory cortex, where we can annotate here, this is an adult surveillance cluster, a control cluster, and we can have here a reactive cluster, which in this case consists of different developmental time points and disease models. And then we can ask, where in this space does the ketamine fall in this? And when we looked initially in the control, it falls into the control. We have then one-time ketamine, which is kind of in the control and a little bit moving out. And we know that here the PNN has not yet been altered so much. And 2 times ketamine, we see that the microglia moving towards the reactive cluster, and we see the PNN already removed. And then at 3 times ketamine, it is truly within the reactive cluster, and the PNN is already mostly gone. And interestingly, if we also look at the recovery process, we see that after 3 times ketamine, and we look after 3 days, that the microglia are going back towards the control cluster. But interestingly, after 1 week or 2 weeks, they decided to go in another turn and go in reaching in an area which is distant from the control, suggesting to us that this repeated exposure of ketamine is not as purely reversible as we are assuming from like general anesthesia. And so we should be having a word of cautions about when we talk about this strategy. And definitely something has been changed in the network, which has been picked up by the microglia in this context. Good, but now looking into, I would like to dive with you a little bit into like this one-time ketamine anesthesia, which has to become a pretty interesting for us, because we started to, we saw like this huge variability, and we also found interesting that there was a difference in the reactivity. Some of them, CD68 was acro-related and some are not. And this morphomics allowed us now to identify that and confirm our suspicion that there is indeed a sex-specific difference. So what we have been doing is here, we have here the control, and we have male 4 hours and 48 hours. So that means we looked 48 hours after the ketamine or 4 hours after the ketamine. There was not so much difference in the morphology, and they all fall within the cluster. However, the female microglia, after 4 hours ketamine, showed a different morphology and recovered within 48 hours, which was like indicating to us there must be some morphological changes that are picked up by the microglia and or neuronal changes that should be existing there, that is being used by the detected by the microglia. So we started to investigate this a little bit more, and using a mouse model in which we have the microglia labeled with CX3CR1 here in green, and pyramidal neurons with labeled with the Taiwan EGFP mouse, and we started to image in the visual cortex. And you can see here, this is a male microglia, and we gave ketamine anesthesia, and there is like, you see the normal movement of the processes going on, but overall not so much differences. Interestingly, when we started to look at the female here, so this is a baseline condition, again, like moving around. However, when we then started to look at the ketamine directly, this is the same recording after ketamine, you see how here the process is like intermingling with this process of the pyramidal neuron in the visual cortex, suggesting to us there must be something different between the male and the female microglia in this response pattern. So which brings me now to the last part, or no, not yet, bring you now to this is just our confirmation, showing you that indeed the female microglia had much more contact with the Taiwan during the recovery process of the ketamine anesthesia than the male. So we quantified the number of the area under the curve, which is significant different between female and male. So at this point, I'm coming to the last part of my talk, where I would like to look into the question about, does sex matter? And you know, and so inspired by this close interaction of the microglia to these neuronal processes, pyramidal neurons, we thought maybe we should just record the synaptic activity of the pyramidal neurons in the layer 2-3 in the visual cortex. So we started to do MEPCs recording 4 hours after the one-time ketamine recording. We did slice recordings and patched the pyramidal neurons and looked at the MEPC amplitude. We didn't see any differences in the female and the male between control and ketamine. There seemed to be no effect at all. Interestingly, when we looked at the frequency, we saw an upregulation or a much higher frequency in the females, whereas the males seem to be rather stable, suggesting to us there is a change in the presynaptic release activity going on within in these pyramidal neurons, but only in females and not in males. And interestingly, when we then repeated this experiment in which we depleted the microglia 3 weeks before, this response completely abolished in females and was not existing, indicating to us indeed that the microglia have to have an impact in these pyramidal neurons to induce this increase in the MEPC's frequency. And inspired by this, we started to look on the effects of ketamine anesthesia on spinogenesis of these neurons. In this case, we used again the Taiwan EGFP mouse in the control, and a male and a female. And the overall number of spines were not different. And then we looked at ketamine anesthesia. Also here, we found an increase in the number of spines in the females. But not in the males. And when we depleted the microglia again, this effect was also abolished and we didn't see an effect going on. This was just specific on one single pyramidal neuron, and we were like wondering, is this effect maybe even broader? Can we say, because let's look again what is overall happening in the brain. And we went back to the microglia, and this was for us also very strikingly so to see. We see when we looked at the CD68 again on the expression, we saw that in certain, almost in all brain regions, we saw an upregulation of CD68. So here in the olfactory bulb, we saw much more CD68 in females than in male. The same is true for the frontal cortex. Here in the dentate gyrus, in the substantia nigra, in the cochlear nucleus, it was not. It was only a trend, and the cerebellum again. So overall, we see that the entire ketamine has an entire effect on the entire brain. So we were like thinking about how would it be if we look at the brain oscillatory network activity to see what is the response going on. And coming back to brain oscillation, also a field I only recently got into it, but find very inspiring in this context, because brain oscillations are described in the brain network as a periodic fluctuation between excitatory neurons and helps to synchronize different signaling features. And you may have heard about EEG recordings that have been associated to certain frequencies. So, for example, the lower frequencies like 0.2 hertz to 3 hertz are associated with sleeping, dreaming, deep relaxation, or resting or relaxed. Whereas then other frequency like beta between 12 and 27 is like alerted and active mind, or a gamma, which I introduced before indirectly, is more on intense focus on problem solving. So of course, this is very simplified view on this, but it gives you a good intuition to think about in this context, how the different networks are coming together. So at this point, we then decided to do recordings and performing tetrad recordings for male and female in animals, adult animals in the visual cortex. And record again from the baseline recording throughout the entire recovery state from the ketamine anesthesia. And as we described before initially, so we give a ketamine anesthesia, and then you see here the massive increase in the gamma range. So you see it, the redder, the higher the power of the intensity is. And then over time here on the x-axis, you see first a reduction in the overall beta activity, and most of the activity going on in the low activity range where the animal is sleeping in the anesthesia. And then slowly you see this recovery in the beta and in the gamma range coming back. And interestingly, if this was now the male I've described before, if you look at the female, you see there is a much higher beta power activity in the recovery process. And you also see that the recovery seems to be much earlier onset in female than it is in males, where it's much later come up. So we can do some analysis in this direction, and in which we subtract the plots and look at the different recoveries. And you can see that, if, for example, in a beta oscillation, the female much faster recover become much faster alerted from the recovery process than the male are doing, and also have a much higher frequency going on. Similarly, we see in the gamma oscillation, initially they are pretty similar between male and female, but then during the recovery process, you see that the female recovering much faster and have an accelerated space recovery from this effect. And at this point, seeing this overall network changes is happening, we got curious what could be the transcriptional signature across all the cell types in the visual cortex. So we collaborated with Bosirka Tasic and did a transcriptional signature from the visual cortex of animals that received either female animals that ever received saline or ketamine anesthesia. And we did single nuclear isolation and performed single cell RNA sequencing. And we found all the cell types that are anticipated to be detectable in this range. And we had to do some very intensive analysis to make sure that we can identify the difference between the ketamine and the control condition. And this is like the data we have seen. So in green is the ketamine. So do we see there's some cell type regions where there is a shift of the population towards the ketamine compared to the control. And we can then look at, for example, the differential expressed genes. And for example, for certain types of neurons, excitatory or the inhibitory neurons, it's very clear that the majority of the genes we found to be changing were downregulated with ketamine anesthesia. So we did this analysis 2 hours after the ketamine anesthesia because we thought the mRNA level should be affected in there. And interesting when we look at the astrocytes and the microglia, which are the ones which have shown the most prominent effect on shifting in the ketamine anesthesia, there we found a couple of genes to be upregulated. And especially one of those genes that caught our attention was FKBP5. And FKBP5, you can see it here, is overall expressed across all the cell types, but has especially high preference in control condition already in the microglia. And when we give ketamine anesthesia, you see a massive shift on the expression level. So almost all of the microglia in the visual cortex seem to upregulate these FKBP5 gene in this context. So at this point, we did then some RNA scope to ensure that we truly have an upregulation of FKBP5 in the animals. So we have used the CX3CR1 probe for detecting the microglia and the FKBP5. This is a male. So you can see there is a couple of dots inside the nucleus. However, not so much as it is in the female microglia, and compared to the control. And you can also, when we do the quantification, we see much more FKBP5 mRNA expression within the female. So suggesting to us indeed that there is an upregulation of FKBP5 specifically in the female context. And we also then looked at the protein level because we never can be sure from mRNA if this is truly also happening on the protein level. And indeed, we can see also that in females, FKBP5 is much higher in microglia in female and less in male. So why FKBP5 is so interesting for us? Because it has been associated in multiple contexts, like depression is like one of the key target genes in depression, and also been associated in a multiplex pathway related to stress, so cortisol level, leading to an association of FKBP5 with heat shock proteins, and then in this way allows the transport of the FKBP5 HSP90 complex into the nucleus, and there allows transcription of genes associated with molecular pathways like NF-kappa B, GSK3 beta, and so on. So there are multiple pathways that can be simultaneously regulated with FKBP5 expression and in the glucocorticoid mediated expression. So based on these results, we were then thinking we should investigate if indeed ketamine induces an upregulation of corticosterone level after 1xKxa. And we decided to look at 2 time points, one at 30 minutes after blood and collected the blood after 30 minutes, so when the animal is in deep anesthesia, and after 2 hours when it's in this recovery, where we also saw this upregulation of the mRNA level. At 30 minutes, there was no difference in the corticosterone level in the blood. So each dot is 1 animal. So we did not collect it continuously, but rather each dot represents 1 animal. And similar is for the ketamine. But here you can clearly see that for females, we have a much higher level of corticosterone in the recovery process, suggesting to us indeed there is a link between like ketamine anesthesia and increase in corticosterone level that leads to an upregulation of FKBP5 in the microglia. And at this point, we started to look into use a selective inhibitor of FKBP5 that's called SAFET2. And SAFET2 has also been used in the context of modulating depression, chronic pain, and obesity. However, it has not made it to the clinical direction because it's a very difficult to dissolve, and it's very, we would say, nasty to dissolve because it contains a lot of like poison and substances, how to dissolve it. So it's not optimal in this context. But still, for our purposes to just downregulate the FKBP5 expression, we thought this might be a good strategy to get a first indication in this direction. So we applied SAFET2 with the ketamine anesthesia. And this was initially the control, so where we saw this massive upregulation of CD68. And when we give SAFET2 with ketamine, we don't see this effect anymore. So we don't see any more CD68 upregulation at all. And similarly, we went back to the morphology and asked, how does the morphology adapt to it when we give like SAFET2 and with the ketamine anesthesia? Would we still see this effect when we do the microglia shift in this shift in morphology? And this is like the morphomics we have been doing now for layer-specific expression of microglia morphology, where just to orientate yourself, we saw that all the microglia within layer 1, 2, 3, 4, 5, 6 are shifting compared to saline condition in females, whereas in males, it rather stays within the control cluster. So there is only very minimum changes to be occurring. And if we then do the same analysis using SAFET2 with the ketamine anesthesia, we don't see anymore this strong separation. The clusters are in a similar range. The ketamine forms like a similar cluster, a morphological cluster, suggesting to us that the microglia are similar responding between male and female. And so at this point, I have, does sex matter for acute anesthetic ketamine exposure? So if you just think about microglia only in females, we saw morphological adaptation, CD68 increase, higher interaction with neuronal processes. And only in female mice, we saw on the neuronal level changes at the presynaptic site, spine number increase. We saw high beta activity, faster recovery of beta and gamma oscillatory activity, and several genes downregulated. And we could nail down that FKBP5, it seems to be an important regulator in this response, and we confirmed the sex-specific differences between male and female. And we could restore certain phenotypes with the SAFET2. And we are now at the final stages to confirm that also on the neuronal level. So at this set, I'm coming to my summary in which I introduced you about ketamine anesthesia, microglia, and plasticity. I talked about how we saw that ketamine reopened the plasticity window, and we are modulating the perineural net. And in this way, it kind of makes the brain plastic to a certain degree again. And we could show that 60 Hertz light entry mimics this effect of the PNN loss. I gave you a short introduction about morphomics and could show you some how we resolve subtle changes in the microglia morphology, which lead us to discover the sex-specific response after acute ketamine exposure, which suggests to us there is a sexual dimorphic microglia response that moderates the visual cortex network activity after ketamine anesthesia. And with this, I would like to acknowledge my team that has been heavily involved in this project, and specifically Alessandro. He has been the main figure on this, and Amin, who helped us with a lot of the programming, and then of course Ryan, who has been involved in the transcriptomic analysis, and Gloria in the morphological analysis. And overall, we have a lot of collaborators. We are always grateful. The IST facilities, who are very helpful for our work. And yeah, if you're wondering where IST is in the outskirts of Vienna, so it's like 18 kilometers from Vienna. This is our campus. This is our where our lab is. And if you're curious to see what we are doing, and there's a lot of different possibilities to come to IST on different level. We have Eastern programs, postdoc programs, graduate school, and so on. And if you have any further questions, I'm happy to answer them.
Okay, thank you, Sandra, for this wonderful talk. We have some questions in the audience. Some of them are very general questions. And I don't know if someone want to ask the question in person, speaking. We can unmute you. Not me, but probably Rosa that has the power.
Yeah, but people can unmute themselves if they want.
Okay.
So, but yes, you can name the people too.
Okay, Guy Brown, he has a question.
Hi, Sandra. That was fantastic talk. But first, I should thank Rosa and Amanda for taking over the webinar series. That's really, really fantastic of you. But Sandra, that was great. I was particularly interested in your first bit about the perineuronal nets and the loss with ketamine and 60 Hertz frequency. I was just a technical question about the WFA, which I think is a leptin which binds N-acetylglucosamine residues. I was just wondering whether that could come about by modifying the sugar residues of the perineuronal nets rather than loss of the nets, because if you lose salic acid and galactose residues, then you would expose N-acetylglucosamine residues. So my question was really, does the loss of WFA staining definitely mean loss of the nets as opposed to sugar residue changes to the nets?
So we have been also applying alternative PNN markers. So we have used acrican and hyaluronic acid, and in this way we also found a reduction of it. So we can be actually pretty sure that there is some remodeling going on. And then also the accumulation of the WFA within the microglia, I think, is also another good indication that it's not just like removing of the sugars in a way, but that the microglia are taken up into their body.
Okay, great. Thanks very much, Sandra.
Okay, I'm going to read out loud some of the questions. There's one, the first one is, please, does CD68 stain resting or activated microglia?
It's usually considered as a reactive microglia. So we usually see an upregulation of CD68 when the microglia becomes active.
Okay, the next one from Wesley Brandau. Great talk. It described that ketamine could block the glutamate receptor, NMDA, and its signaling is important for microglia to do the synapse pruning. Do you know if it is possible to reproduce the phenomenon that you showed using another NMDA blocker such as other NMDA blockers as MK801, or if you have a different response to ketamine in the animal's maturation period? This is P1 to P15.
Mm-hmm. So we have not yet considered to look at MK801. We know, yeah, ketamine itself is like a tricky drug itself. There is a lot of like debate on the mechanism of it there. We know that it has some NMDA effect, but there could be also alternative possibilities in there. And there is a very much dosage dependent effect in there. So we were very pragmatic at that point, was truly just to anesthetize the animal, make sure that the animal all are anesthetized, and then look what the effect is. So that was our initial intention. Yeah, so we have a clear behavior readout, we have a clear response, which we have seen is like the PNN loss. So now we could think about, as suggested, we could look into the MK801 or alternative strategy. What we know is like when we used alternative anesthesia, for example, isoflurane, which acts on another mechanism. So it's not like on NMDA, it's acting on GABA receptor directly. There we didn't see this effect. So there seems to be some combination on this ketamine that we should keep an eye on. And yeah.
Okay, then Carlos Matute has a question. I don't know, Carlos, if you're there, maybe you can connect yourself. Which enantiomer did you use in your experiment? SR or a racemic mixture?
Racemic mixture. Oh gosh.
Okay, there's another one. Did you use 40X or 100X to study the morphology of microglia?
I'm sorry.
The objective.
40X. 40X.
40X. Carlos has another question. Maybe I don't know if Carlos, you have another question.
Yeah, hi Sandra. Nice data. Congratulations. So I am one. I mean, as you know, is ketamine. Ketamine is a antidepressant, which is now very fashionable in the clinic. So yeah. I wonder whether the efficacy, the therapeutic efficacy, has also a sexual dimorphism. So, and the reason why I ask you this is because it looks to me like the ketamine is just making the brain more plastic in general. So perhaps what it's doing in depression is rewiring part of the circuitry, and then that's why depression is overcome somehow. So I wonder whether there's any gender dimorphism or whether you see any parallelism between the studies, the beautiful studies you just told us today on the visual system with the disease itself.
Mm-hmm. So, yes, it has a very interesting link with depression, and that's something we were very curious about with the 60 Hertz in this context. If you could like recapitulate this. Related to ketamine, we have done some experiments in which we got access to human temporal lobe tissue fresh from surgery, and we exposed it to ketamine directly. We got male and female samples and just exposed it to ketamine in high dosage. And there we also found a similar effect, that the microglia got reactive and started to remove the perineural nets, but only in females again. So suggesting to us there must be some sex-specific differences going on the direct effect on ketamine. I'm very curious to get to know if the data related to depression and treatment, and if there's sex-specific difference between male and females. I mean, I think there are studies now ongoing with the treatment exposure now in citizen therapeutics, but I haven't yet seen any yet coming out. So I'm very curious to see if there is a known effect between male and female in this context.
Just a small curiosity. So in your stainings with the Wisteria floribunda, I love this name, agglutinin, there is plenty of staining in the white matter. So I wonder what is, it's just very peculiar to me. And this is removed also when you use ketamine, goes away. So I don't know whether you have any thoughts about that. That is very curious to me.
I honestly don't know the data in there. We haven't looked into this as with in the white matter extensively there.
It goes away in your slides. It was very clear.
It was okay, but we have never really quantified this because we wanted to be. We have to be sure. We usually, most of the time, the people are doing like intensity measurements where we will always feel like this is suboptimal. So for us, we were more focusing on identifying the coded PNN structure because this is very clearly defined. We can see very clearly the neurons encapsulated and when it's gone. That's why I'm a little bit hesitant to say now it's completely gone. Yeah, but I have no thoughts about this. Yeah, but it's
okay, thank you.
We keep it in mind.
Thank you. Congratulations for your work.
Thank you.
Okay, I have a very interesting question from Kenyin Chu. Very fascinating talk. Do you think it is a general phenomenon that activated microglia destroyed the PNNs? Do you also see a similar PNN loss effect when microglia are stimulated with, for example, LPS?
Okay, we have not yet done LPS stimulation. LPS is a very, if I hypothesize, it's a very general systemic inflammation, could induce stress. I'm not so sure. I think it has more to, I would probably not expect to see changes because I think that this is specifically with the neuronal activity going on that you have like this this drop of the neuronal activity with the anesthesia and the system reinstate. That means you have to imagine that your system is down and you have to restart. It's like you start turning on your car in the winter or so, and it's like you have to. The brain has to get back its synchronization, and I think that is the critical part, where the neuron sense close to paravalvular neurons that something is off, and that they start to modulate here. So we are now looking also like in modeling a neuronal activity related to ketamine, and we are with collaborators, and so we try to identify if there is how this comes together, this neuronal activity with the ketamine. With LPS, I would not expect it, but I might be wrong.
Okay, thanks. Now there is one from Carlos Diaz Salazar. Maybe you can unmute and make yourself your question. Carlos?
Thank you so much, Sandra. It was fascinating. I had a question that was kind of similar to Dr. Carlos Matute. It was related to the repeated ketamine exposure that with the molecular deprivation model, you could observe a more permissive or plastic state. I was wondering if you've tried to investigate that further and look at if there's an improvement in behavioral tests that are normally associated with juvenile mice or tests that require a not yet closed critical period. Have you tried some of those learning tasks? And if so, do you think there's a transient would be a transient effect, as you saw with the molecular deprivation model, or would be a more permanent effect?
It's a very interesting question, and we were thinking about what would be a good behavior paradigm, because the challenge is that this paradigm takes a week. That means how would you design the behavior paradigm that the animal learns, and which part of the learning would you do? And we had some thoughts. We were trying out novel object recognition, but that was not so optimal. That was also during the phase of COVID and lockdown, so it might have been a little bit messed up also there in that content. The question is easy, the answer is not straightforward. So if you have an idea about a good behavior paradigm, we would be very happy to discuss it with you.
Okay, I have two more, and then I would like to ask a question myself. This one is from Daniel Griffiths. Does ketamine increase activity only during periods of anesthesia sleep, or does it override beta-adrenergic inhibition of microglia to increase microglia activity?
So the first question was about sleep. Sorry.
Yeah, that's ketamine increases activity only during periods of anesthesia or sleep, or does it override beta-adrenergic inhibition of microglia to increase microglia activity? It's a mixed question.
Yeah, I think it's an interesting thought, an interesting perspective about it, because I have a little bit the idea behind it that there's probably this two-strike model, I think, because I think that there must be something behind it, because the repeated exposure. I think because the first time you don't see it, the second time suddenly it happens, and the third time it's really like from day to night. It is like a really massive. So there must be some adaptation going on. If it's now like this proposed mechanism that is suggested there, could be. I mean, we still, I mean, we have all this the transcriptome analysis of all the other cell types, and we have not even started to touch that one. We could see like what are the transcriptional signatures on the genes itself, how receptors is there differences in the cell-cell interaction, and we also even not have included the astrocytes in this whole game, which is another level of complexity, because astrocytes seems to also respond on this one. So I cannot give yet any satisfying answer, but yeah.
The last one. This one is an obvious question. Thank you for the interesting talk. Did you check whether the sexual dimorphic microglia is a matter of hormonal cues? This one is from Evangelia Kyriakidou. So did you check whether the sexual dimorphic microglia is a matter of hormonal cues?
Okay, hormones. Well, we are a little bit debating about his hormone things with morphology. So in the paper from Colombo and Cubero, what we have published, we did also overrectomized animals, and then looked at the morphology in adult animals. And we see a complete different microglia morphology, which neither reflects a female or male or anything. So it seems like if you completely deplete the hormones, something has changed in the microglia morphology shape. Is this morphology what we see now with the ketamine just hormonal driven? I don't think so necessarily. I think it's more like probably a stress response what we see based on our data which we have with the corticosteroid and the FKBP5 expression level. So I think in this line, but yeah.
Okay, I have a couple of, there's another one before mine. Could you elaborate on the astrocytic response to ketamine? Maybe this topic.
Shouldn't have said it. No, we haven't looked in detail in it. We just see that the astrocytes also upregulated several genes, and there seems to be some effect there, which I'm not yet an astrocyte person, and we look if somebody is interested to look in the astrocytes, we're more welcome to share the data.
Okay, my question, this was the last one, and my question has to do with the matrix, which is the topic I work in. And you know that the perineuronal nets, they are a very important part of the matrix, but they are only surrounding moving neurons mostly, and there is much more matrix than the PNNs, I want to say. And so have you checked if microglia with your treatment of ketamine, of repeated ketamine, they increase phagocytosis of other matrix components? Because, for example, around glutamatergic neurons, for example, there are other matrix components such as hyaluronan or other proteins, and maybe they are affected as well, not only the PNNs.
No, I would not exclude that. I think it was one of my grants which got rejected, so I think that's why we didn't look for. We didn't have the support from the community to look further into different compositions on that. But yeah, I agree. There is much more matrix molecules to look at, and it would be curious to see if microglia have a certain preferences for certain matrix compositions. I mean, I'm still amazed about this difference between the 40 and the 60 hertz and the selectivity, which seems to, in both, in 40 hertz, the amyloid plaque, and the 60 hertz, the ECM or the perineural net. So yeah. So this is something definitely curious to further follow up on that one.
And the last one, just this one is short, but regarding the question from Daniel Griffiths about the sleep. You know, when you sleep, the extracellular space gets bigger, and this is the same that happens with ketamine. It's true that this is with one anesthesia cycle, not with repeated like you did, but maybe with ketamine, when the extracellular space is bigger, maybe microglia has more access to the matrix, so it's easier for them to degrade it, or this is too far-fetched to?
I didn't know that it's getting bigger, but yeah, could be a potential thought. I don't know.
It was a famous paper by Meike Niedergard. She showed that the ketamine does to the extracellular space, ketamine silencing, that's the same as the sleep. So when we sleep, the extracellular space gets larger and removes like.
Okay.
So maybe, I don't know, maybe it's something to do with this, with the ECS.
We'll take a look at it. We'll see. Thanks.
Okay, I think there are no more questions. I don't know, Rosa, if you want to close the session. And thank you, Sandra, for your wonderful talk.
Yeah, exactly. I just want to thank you again, Sandra. Thanks everybody in the audience. And well, see you next month, the 6th of November, you will with Dr. Francesca Peri, and you will receive all the information, further details by email and by Twitter. Thanks again to everybody. Bye.
Bye.